Susie,
Rob.
what do you know about life in the Devonian?
Lots of fish. There's lots of fish in the Devonian, right? That's the time
A
of
lot of fish.
fish.
Age of fishes is classically what we call it. Yep.
Yeah. Not much happening on land. Earliest plants? Are they a bit earlier than
Yep.
that?
Yeah. Let's move on to land, though. I guess we think of the Devonian as the age of fish, but what's happening on land? From someone like you, from a dinosaur, paleo, not, Mesozoic perspective, what do you think about Devonian life on land?
I probably very little. Probably very
Yeah.
little happening on land. Maybe the earliest plants, maybe some insects. Don't really know. Not really got a clue.
Well, you've nailed it.
Oh,
There are
oh,
some
good.
plants. There are some plants. There are some insects on land. But there were also these gigantic, mysterious, potential fungi type things called Prototaxites looming over the landscape, 8 meters tall, 1 meter wide, of completely dubious, weird provenance, which is what we're going to get into today.
I'm very excited about this because I've been saying to you since this paper came out, Rob, I want
Yeah.
to do this Prototaxites paper. I don't know anything
Yes.
about these, and they look really funny
Yes.
and a little bit like penises. So we should do this on the Fossil Files podcast.
How much was this motivated by scientific curiosity or childish whims?
I'm going to go 50-50.
Okay, so let's find out about these giant phallic, not fungi things today. So, Susie, what have you been up to? It's been a while since we last spoke.
I know it feels like forever since I last spoke to you, Rob.
Hmm.
Actually, I have been away for the last 3.5 weeks. Was two
Hmm.
weeks doing fieldwork in Morocco,
Hmm.
which I know we're going to talk about, aren't we? As a little bit of
Yes.
content for our subscribers.
Everything's content. Everything's content. No, I look forward to hearing more about your Morocco adventures and what you found and how you got on. But we're going to do that as a separate, dedicated episode coming up next. What about outside? Well, I guess that's probably taken up all your time, right? Going to Morocco, that's
Well,
quite a big adventure.
I did manage to squeeze in a short holiday to a geopark in southern Spain, which was very lovely,
Oh,
called
blimey.
Gata. Yeah, it was beautiful. It was very, very nice. And
Where in
also,
Spain is that?
it's down southeast Spain, so it's the Betic Cordillera. Many people will know it because every geology undergrad goes there in the first or second year. But it is also actually an incredibly lovely, very wild. It's all volcanoes and it's kind of amazing and beautiful beaches and clear seas
So,
and very
just to
nice.
be clear, for your holiday, you went to a geopark.
Yeah, I mean, I am married to a geologist. It is basically what we do. Our
Right.
daughter is not that keen on it. It has to be said. She's 13, and when we make her go and look at rocks, she gets exceptionally grumpy. So she had a bad day when we made her walk around an old gold mine. But most of the time we let her snorkel on the beach, so it's fine.
Kids, let's go to the abandoned gold mine. Excellent. So, I've
Anyway,
also been away. I've been on holiday to Greece. So, on the
have
day
you?
that our intelligent giant octopus episode came out, I was busy stuffing my face with octopus. I thought that was the right thing to do.
Then my
Do
whole spiel at the beginning about how we definitely
Yeah,
shouldn't be eating octopus.
but they shouldn't taste so good. That's the problem. Apologies to everybody, the big octopus fans out there, but they taste good and they are intelligent. One day maybe I should stop eating them because they are too clever. But I don't know. We don't have that same conversation about pigs. Pigs are very clever.
I know. I
Maybe
was
we
thinking that
okay.
actually. Yeah. Well,
Well, we know
I
what's
don't really eat
coming.
that. Well, I eat bacon to be fair. Yeah.
We should
Anyway,
stop eating meat altogether, shouldn't we? Really. Anyway, that's not what this episode's meant to be about.
can I
For
just say
this
though, Rob?
episode, yes.
Last episode was cracking. I
Mhm.
listened to it last night. I went did
Oh
a bit
yeah,
of field work on the Isle of Wight the last couple of days, and I was
yeah.
driving home from Portsmouth, and I did listen to it, and I thought it was very funny. So I would say if you haven't listened to the giant octopus episode, you should absolutely do so
Check it
because
out.
it's a
Is that
bit
no?
of a good one.
Is that a weird experience listening to you and I in your own car, listening back?
Do you not do that? I mean,
No,
obviously,
I don't. I don't.
I have an absolute monster ego, like most academics, Rob, and love listening to myself.
Well, I guess it's different. I have to listen back to them to edit them, whereas I guess you don't. So I guess if you want to find out what we actually, yeah, okay, it makes sense. It's different for you. For me, I'd have to be listening to it for like too many times.
28th
But
time.
yeah,
Well, I've often forgotten what we've said and what we did and what we talked about. I have to listen to them again so I can remember the papers. It's like
well,
I'm experiencing it all over again.
I also have to edit out your most libelous comments sometimes. But I don't think there'll be any libel today. So let's talk about Prototaxites then. So this week I've chosen a paper. Although actually, no, I haven't. It's one that you've been asking for a while. You've been prompting me to do this one. So
I have.
it's time.
I've been campaigning for this one. However, I haven't actually read it. So I am delighted. Well, I did only get back from the Isle of Wight yesterday, and you actually only sent it to me yesterday as well, Rob. So
Yes,
yesterday
that's
afternoon.
true. That's true. Yeah.
But I would I'm actually really delighted because not only do I really wanted to know about this paper, I don't have to read it. You're going to tell me all about it, so it's even better. It's a win for me, I think.
It's making life much, much easier. That's true. Okay, so this week's paper, unusually for us, it actually came out about 6 months ago. Normally we try and do things much more recent than this, but we've got waylaid by other things. This was published in Science Advances in January of this year by a team from the University of Edinburgh, so a wide range of different people there. This is Prototaxites fossils are structurally and chemically distinct from extinct and extant fungi. So this is about one of the really interesting paleontological enigmas that's been going on for a long time, which is Prototaxites, which was these giant columns, phallic columns if you want to, that littered the terrestrial landscapes of the Devonian and late Silurian. So they're weird, right? They're massive. They are, I mean,
how big are they?
8 meters tall at the biggest and about 1 meter wide. So
That's
big. We're talking big here, but they weren't all that big. But, obviously with these things, as evidenced by the octopus episode, you've got to report the biggest sizes to make it more attractive.
But Rob, how are they preserved? I mean, apologies if you're going to cover this. How are they found as fossils when they're they're 8 meters tall? Are they like trees? So they sort of end up lying down?
Yes, and that's going to become part of this story later on. Yeah, how they end up getting preserved is quite important, especially because we're going to start talking about the Rhynie Chert, where things are weird and unusual. Yes. So we're talking in the Devonian. So as Susie said in the intro, that's the age of fishes, right? Classically, that's where I do a lot of my fossils that I work on come from the Devonian. It's the best time in the whole world. Actually, that's not entirely true. The Silurian's better. But the point is, the Silurian and the Devonian are where the action's happening. But most of that action is happening in the sea. On land, we're dealing with the early colonizers on land. Often the forests, we'll have this period of time called moss forests. So we've got Cooksonia, which is about, I don't know, a centimetre or two big, and it looks a bit like a moss, but it's not a moss, and it's all over this. We'd have a green carpet on land. Not a lot of animals on land. Maybe a handful of centipedes, insects, spiders. Not in the Silurian. This is only now in the Devonian when we've got this sort of thing happening. Although maybe in the... Well, okay, that's not true. We're going back to the Ordovician, maybe. Too much detail. The point is, on land, we've got these nice mossy forests, and then we'd have had these looming, weird, massive columns over the landscape, these Prototaxites. So they've been known about since the 1800s, and there's been a lot of debate about them. So since about 2001, we've come to the conclusion that they were these giant fungi. And in some senses, that makes sense, because they've got all these tubes packed inside them.
So what did people think they were before?
Well, that's the problem. They're enigmatic. And there's arguments about what they could be. They seem to have these large columns. There's some branching structures. They're packed full of tubes. So what are they becomes quite difficult, because they don't have a lot of morphology. They don't have the characteristic chemicals of something that's photosynthesizing. So that's number one. So they're not a plant, but also it's too weird to be an animal that like this. So fungi is a natural candidate because they seem to have some sort of elements of being large and multicellular, and people were thinking, well, maybe it's some sort of chitinous type thing, and it's just a massive fungus. Now fitting it into a particular part of the fungi has been tricky, but that was the best bet of what they were. And also, because this is happening before forests existed, that makes it so the Devonian terrestrial landscape would look really weird, right? It wouldn't have looked at trees and forests just only started turning up in the late Devonian. So in the middle of the Devonian, you'd have had this weird looking mossy forest with the weird columns looming above it. So that's the context of where Prototaxites is, as this really weird thing that a side note, I was looking at why it's called Prototaxites, because someone originally thought it was a fern who discovered it in the 1800s. So proto means sort of beginning or sort of almost early, and taxites means yew, like a yew tree. So but then someone came along and said, actually, I think it's a fungus and I think we should rename it. And then people kicked off and said, no, you can't rename it just because you think it might be something else. That's not how taxonomy works. You have to stick to the original name.
It's true.
Yeah. So it sticks with this name, even though someone's calling it an early yew tree, and we both all agree that it's not a yew tree. I guess that relates to the Oculodentavis episode where we looked at that dinosaur that wasn't a dinosaur. It's called a big-eyed bird which didn't necessarily have teeth and wasn't a dinosaur. So that one, well, oh, bless you.
Thank you. Yeah, there's also a dinosaur called Micropachycephalosaurus, famous for the fact that it has the longest dinosaur name. And it isn't anything to do with Pachycephalosaurus either. It's not even a pachycephalosaur.
So I guess you've got to be careful with your taxonomy. If you're going to go and calling something using another name, like Nanotyrannus and Tyrannus, well, no, let's not get into that. That's a whole long story that would exhaust me. And we covered it last time in our episodes in November when we went to SVP. Anyway, this was the context of what we're dealing with. This paper is now introduced. So what these guys did was want to go and try and solve this problem by looking at some Prototaxites specimens from the Rhynie Chert. Have you heard of the Rhynie Chert before?
I have. Yes, it's very famous.
Yeah. And what do you
do you want me to expand on that?
I was hoping you might expand on that. Yeah.
Okay, so it is. It's from just a couple of sites in Scotland, isn't it? And it's I think, is it a silicious spring deposit? Is it something like that?
That's right,
so you basically have incredible preservation of very, very fine details of the things that were living on land at the time. And so all of these plants are, and things like fungi and things with spores. I went to a great talk at the Natural History Museum the other day with somebody who was looking at these things, spores and things like that on these. And they're preserved at incredible cellular level detail.
Yes,
and
absolutely.
when they're very tiny, in 3D as well. So they were doing the talk I went to. They were getting some really using some really super cool microscopy techniques to get kind of 3D structures out as well. So yeah, it's really important, isn't it? It's like it's sort of critical for our understanding of early Devonian land plants.
Absolutely. So Rhynie Chert is, as you said, from this really weird place. Well, it's not weird. It's Aberdeen is quite a normal place, but out in rural Aberdeenshire, there's this layer that's sort of hidden under the ground under about 3 or 4 meters of overburden.
It's basically in a field, isn't it? As far as I understand.
Yeah. So it's a bit difficult to access, and it's also a protected site, so you can't just go and dig stuff up there. But it's also really unusual for a fossil deposit. So I do a whole bit of my undergraduate teaching about fossil Lagerstadt, and we talk about this one good, but it's also weird because it's not really representative of other ones, right? So it's a hot spring is what it is. So we've got a sort of volcanic environment with hot springs, and everything in it has been silified, turned into silica. So that's unusual. We don't get this kind of fossil preservation very often. So it's a bit like Yellowstone National Park in Wyoming or whatever. You've got this sort of really weird chemistry, and things get turned into stone in an unusual way. But that allows you to preserve the really fine 3-dimensional subcellular detail. So things that the Rhynie Chert is famous for. Some of the earliest plants, it's got insects. So this is some of the earliest land things. Insects that look like they might have been flying. We've got some trigonotarbids, which are these
what are they?
type things. I was just explaining. They are arachnid type things, chelicerate type things. The creepy crawly spidery type things. They're not spider. Well, Russell will tell me off about it. My colleague in Manchester here works on these, and he'll tell me off for getting it wrong. But they are. They look like spiders, but they're not spiders. And what else is there? Yeah, fungi. There's also fungi there, like known fungi, which is unusual because we talked about this before. We've ended up talking about fungus quite a lot, which has surprised me a little bit, but very rarely does fungus end up part of the fossil record, but it does there. And bacteria. They even have bacteria preserved there because you've got this fine subcellular detail. So that's what's going on in the Rhynie Chert. So they took this specimen or sample from a 5 centimeter block of it from there and subjected it to some nice high detailed analyses. So they took some nice thin sections and they tried to reconstruct a nice 3D model of what the morphology inside looked like. Basically, it's a whole bunch of tubes. Do you want to describe to the listeners what? Have you got the paper in front of you, Celia?
I have actually, yeah. Oh yeah.
Can you describe to us? Yeah, what's happening in Figure 2?
Well, there are a series of panels which are, I haven't read the caption, but they're microscope slides, and they've used a method called CLSM, which means nobody knows.
Were you expecting me to fill that in? I don't know.
I haven't read the paper, dude. She only sent it to me last night.
Confocal laser scanning microscopy.
Confocal laser scanning microscopy, which shows that you've got these kind of like series of tubes through these things, and then they've reconstructed them in 3D. I guess because the thin section, although it's very, very thin, these things are very tiny because they're cellular level, so you can get kind of a 3D structure from it. And it's like an anastomosing series of kind of tubes
Yeah. So
with some blue
when you say
dots.
anastomosing, let's remember that anastomosing might not be a phrase that most people are familiar with.
What? Okay, that's sort
Come
of
on.
branching and rejoining again.
There we go. Branching and rejoining. That's absolutely right. Did you genuinely think that anastomosing was a
Anastomosing
phrase
is
that people would use in day-to-day parlance?
yes,
No way.
it is. Crashing on.
Crashing on. So we got. Sorry, you're quite upset about that. So I have to be careful about here. Then we got a series of branching tubes. It's basically it's really really packed with different tubes of different lengths and orientations. Okay, that's part number one. So they've highlighted the morphology, but some of that was known already. But the second part is that they did a load of chemistry on it to analyze it. So they did some FTIR, which is, oh God, I have to remember the top of my head. I've used the technique myself and I still can't remember off the top of my head what it actually stands for. Fourier transform infrared microspectroscopy. So you are going to be blasting the specimen with some infrared, and you'll be using that to try and detect what the sort of organic signature of things inside it. So the particular kinds of organic chemical bonds in it will have a particular kind of signal, and you can detect what's going on inside it. You happy with that?
Yes, although I have questions, but carry on.
Go for it.
Like maybe you're going to address them.
No, this is the right time to ask a question.
Okay. So you have mentioned how the Rhynie chert has this very unique chemical signature where it's caused by hot springs and everything's replaced.
Mm-hmm.
How are we getting the primary chemistry from it?
Well, there are organics in it. So they do talk about this, and ordinarily I worry about this sort of thing because if you're doing these sorts of analyses of the chemistry at a fossil, how much are you picking up the chemistry of the original fossil, and how much are you picking up the alteration at that particular site? Like, right. If I was to compare Mason Creek with Jehol biota with Rhynie chert, I'm going to be picking up the chemistry of the rocks and the environment that thing found itself in and all the effects that come from it. But what they've done is they've compared only fossils from the Rhynie chert.
Okay.
So I think that makes life a lot. I think that neatly sidesteps that whole issue.
It does.
Because they're comparing things within the same place, so you would expect them all to have the same history of taphonomy and diagenesis. So,
I mean, if the chemistry that you're finding is entirely due to the preservation, they should all have the same chemistry. But if they have different chemistry, then that suggests that's something inherent to the animals, the chemistry, and the plants and the fungus and everything.
Indeed.
Organisms.
Now, there are some circumstances where you might say, well, given the nature of chitin, that chitin might have had a different track record to what would have gone in comparison to, say, melanin or whatever. But as you pointed out, if it's the same thing in the same organism at the same site, then maybe it should be detectable. So they then analyze the chemistry of Prototaxites using FTIR, using a number of different things, synchrotron ones, desktop ones, other kinds of ones, and they put into analysis with plants, cyanobacteria, arthropods, bona fide fungi, and oomocetes and amoebae. So these are all from the Rhynie cherts, and they've all been analysed using the same technique. And they sort of map it out, and they train some models to sort of identify it. So first of all, Prototaxites sits well outside the rest of everything else. Like it's a long way from the bacteria, a long way from the fungi, a long way from the arthropods. It doesn't have the characteristic bits of chitin in it that you would expect, because chitin is a big deal for fungi, a big deal for arthropods as well. You have to have this particular kind of big... Is it a carbohydrate? I can't remember off the top of my head. This big sort of structure that would be easily detectable, or should be detectable, having a specific signature from the... it's a polysaccharide. So they don't have it. So Prototaxites sits outside of this. That's kind of the headline, is that this is weird. Prototaxites is not this, it's something else. So they also did some training of these models. So they then said, right, okay, let's train a model to identify when something is or isn't fungi. And they said, no, Prototaxites is something else. Then they did, let's train the model to identify something is and isn't plants, and no, Prototaxites is not that. Train a model to identify when it's bacteria, is it isn't that, and it isn't. Prototaxites is consistently being identified as something else. It's weird.
It's an alien.
So it's an... I was wondering how long it would take before we ended up with it being an alien. So they go through this long process of ruling out what it isn't. So it's not a plant. It's not an animal. We should have been talking about the sorts of fungi. What a characteristic of it being a fungi? What would that mean? It being a large saprophytic, so that's eating the rotting matter, makes sense. Maybe it's just some sort of fungi. There was also debate about whether
quite
it could be
big,
some.
wouldn't it?
It would be big.
It's quite
but if
big
there's
to
nothing...
eat just some mossy things.
Yes. But it also might have had enormous hyphae stretching off in all sorts of different directions. We don't know about that because all we've got is this large column of a thing.
Right.
But there might have been a big root of mycelium and hyphae going off that we have no idea about. So that it could have stretched out for miles. And indeed, that's what some fungi do that sort of thing today. So there could be a whole missing part of this that we don't know. And also maybe it was a lichen. Maybe it was undergoing some sort of photosynthesis. Maybe it had some algae embedded in it, and that maybe explained why it got so big. And, I came across the word lichenized in here. So it might not be a lichen like it is today, but it might have been something a bit like that.
So like a symbiont of some form.
Yeah, some sort of symbiont is a possibility. And I think that was quite a reasonable hypothesis until this came along. But they said that they're looking at all the morphology and the chemistry, they rule out any kind of symbiosis. They think it's packed together with just one thing. So, and it also rules out it being also the morphology rules it out being some sort of fungus. So these structures are consistent with functions in transport and exchange of substances without parallels in extant fungi. Such physiological functions could have played a critical role in allowing Prototaxites to reach large, freestanding sizes. So, we reached the conclusion with this big detailed analysis. It's not got the morphology. It's not a symbiont. We know that. And its chemical signature, and together all the pictures indicate that it's not a fungi, it's not a plant, it's not an animal, it's something else. So what the hell is this thing?
This is super cool. What is it? Do they make suggestions?
They're quite cautious in this paper. They actually spend quite a long time talking about why their data might be wrong, and I quite like that. They talk about all the factors about why it might. I mean, it's possible that reviewers made them say that, but it didn't come across like that. It came across like, here's our long section of all the caveats they're applying to this.
Well, that's good. That's what you should be doing.
Which
Error
I
bars.
Them for.
You should introduce them to the octopus guys.
I haven't heard from the octopus guys after that. I don't know if they've heard. But yes, this seems so. They've definitely thought about the different kinds of chemistry and what could have happened, and whether some things were happening in one sort of thing or another place or whatnot, and what the artifacts of the way that they use the techniques. But it leaves they're a bit cautious about speculating about what it could be. But one of the implications of this is that there's some other unknown, unique life form out there. So large multicellular things. There's us, the epistoconts, which is fungi and animals. We are part of one group. Plants have done it. Algae have done it. Red algae, I mean. Yeah, red algae. They've done it separately from the green plants and the algae.
What, made really big things? Or just multicellular things?
Big multicellular things.
Okay.
So if you think of like the kelp forests, are they...
Oh, yeah. Okay, yeah. Fair.
I don't actually know if they're red algae or not. But the point is, I can't... Before I get bogged down in algal taxonomy, which...
Okay, sorry.
might be a bit beyond what I can address. The point is about... This thing is not like those. It's not an algae, it's not a plant, and it's not an animal fungus thing. So it's something else. What is it? Is there some enormous third branch of life that we don't know about?
This is super cool. It's super cool, Rob. It's an alien. Or is it an alien?
So now it's time for us to speculate. So they obviously, as authors in the paper, can't speculate. We can. Well, you've speculated.
How can we speculate about something that, like, I mean, you can't. It's really difficult to speculate about something that you don't know, right? How can you speculate about a life form that we don't know anything about? That doesn't. We haven't. I mean, it's very exciting. What did it do? Why did it need to be so big? What was it doing that made it in a landscape where everything is no more than a centimetre high? Why does it need to be 8 meters tall? Is it a landing structure for an alien spacecraft?
Well, this is why one of the reasons
I think it is.
I was just about to say something serious and then you distracted me with the landing.
Are you suggesting that's not a serious suggestion?
Yes I am,
okay,
Susie. So that was one of the reasons that it might have supported its fungi idea, because fungi, transmit their spores, right? So if you are very tall and you've got your fruiting body at the top, then your spores can be distributed very, very far and wide, and evolution would rapidly feel you to that conclusion, one assumes. Although I don't know why fungi aren't enormously tall nowadays.
And plants, they also need to distribute pollen, or I don't know whether these mossy type animals, what were they called? Cooksoneas. They had some sort of spores that they needed to distribute, and they only needed to be a centimetre high to manage it.
Yeah, that's true. So maybe that
8
line of
meters.
thought. Yes, at
It's
their
tall.
highest. Well, side note, I said I was going to come back to this. There was some debate about whether or not they are actually upright. They could have been lying down.
Oh.
But that to me seems even more silly. Why would you need to be a big chunky log lying down? That doesn't make any sense. Things
No.
that do that nowadays have like long networks that have to do that. Having a large thick column with lots of tubes in it makes sense if you have to be upright for some reason.
Upright. Yeah. Interesting.
So, was there something that wasn't animals, wasn't fungi, wasn't plants, and is some other big multicellular eukaryotic thing going on out there?
But okay, so we've had a conversation before about what things need to be alive, like what the definitions are. I mean, I guess the most fundamental aspect of living is reproducing, right? You have to be able to reproduce yourself.
Yes.
So the thing about having a fruiting body at the top and needing to be taught to get your spores out could still be true for a different form of life, like a different multicellular life form. It's still going to have to reproduce somehow, and it probably would have done so via some sort of spore, because that's kind of, I guess, relatively basal. Would it be? Would you say that?
Yes. I mean,
most
I guess
early diverging things.
Yeah, having spores is not unusual. I guess that's the point. Yeah.
I like Figure 4, where basically they've gone through and they've said Prototaxites features, and then they've talked about the taxonomic groups that you can exclude based on the features that it has. So, for example, it says complex multicellularity excludes prokaryotes and oomycetes. And then they have cell walls, so that means it can't be an animal. It has a tube-like morphology, which means it can't be a brown alga, and so on and so forth. So, and at the end, they've got a whole list of things that it has that are not present in fungi, and their conclusion is no affinity with known complex multicellular eukaryotes. That's badass. Why did this paper get in Science? Why did the octopus paper get in Science and this one didn't? I bet they submitted it to Science. They
might have done. I think I would have done if I was there.
Oh, definitely.
So I think they've done a really good job here. I think, as you say, of stepwise going through and eliminating things. So let me find the quote from the end of the paper here. So, with no support for fungal affinity, we suggest that Prototaxites is best considered a member of a previously undescribed, independent and extinct lineage of complex multicellular eukaryotes. Our results also directly support previous investigations of the molecular composition, suggesting a possible structural composition of blah, 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 blah. So basically, it's something else. It's some other weird, unknown thing, and that's super cool.
That is super cool. And it could be a landing port for an alien spacecraft.
Well, they don't go out of their way to say that it isn't that. But this comes into a bigger problem, right? I've got into this problem when I went into the world of Tully Monster, which that's a long story for another time, I guess. But that was I had a foray into that world. So Tully Monster. I guess I'm going to have to now explain this now. For those
we should do a bonus episode on the Tully Monster.
No,
we could get Thomas Clements in and get him to talk about the Tully Monster.
Well, no, because he and I ended up on different sides of that debate.
Exactly.
So for those that aren't aware, Tully Monster is a weird, unusual thing from the Mason Creek in the Carboniferous. But there's lots of debate about what it might be. But it's quite difficult when you end up in academic debates about what something isn't. It's because that's not actually very good. It's better to have a hypothesis of what something is so you can test that. These guys haven't speculated about it. They've just said it's something else new. We'll have to save the Tully Monster chat for another day.
It's very difficult to hypothesize about something where what hypotheses could you set up about, is there a way that they could have come up with something? I mean, it's a bit difficult, isn't it? Because you have no baseline, you have no. So, I mean, we could go through, as I say, the things that we know that life forms need to be able to do. They need to be able to reproduce. They need to be able to feed in some way. What other things they need to do? That's probably it, really, isn't it? So, and how it might have been doing those things. But I don't know. I mean, I guess that's a way to get it. Yeah, that's the only hypotheses we have. We have nothing else, do we? So I think it's quite difficult.
So I guess they don't really raise a hypothesis. What they're saying is they're overturning a hypothesis. And I guess that's what science is about, right? Someone else has raised a hypothesis that it's fungus. They've overturned it with a very detailed analysis about what's going on. And they've been quite, caveat about here the things that they've done. So I like that. That's good. That's good science. So we're going to give them a... I mean, I'm not quite... in the position to analyze exactly what they've done with all their modeling and whatnot and the chemistry underneath it, but it seems to be sensible to me. So that's the story.
Very good. Well, I'm very
What
excited.
do you think as a take-home messages? Oh, I did have a separate final 5 minutes of extra dinosaur things for you because we can't leave it there.
We can't only talk about literally a new form of life because that's not exciting enough. I think this is super cool, and I'm really excited by it. And I think I don't know that there needs to be a take-home other than, oh my God, the fossil record has the potential still after all our exploration. I mean, how long's the Rhynie chert been known about? 200 years?
Oh, a long time.
200, a huge number of years to still discover. And I'm excited. People say, "Oh, do you think there's more dinosaurs to discover out there? Are there new species to discover?" There's not just new species. There's an entire new kingdom of extracellular, of multicellular life that we've never seen that is out there. I think that's incredible.
It is incredible. Just as a extra bit of context, Prototaxites isn't just known from Rhynie chert. It's known from lots of different places.
Is it?
But the Rhynie chert, it's known from lots of places. But the Rhynie chert gives you that extra window of really good preservation. So this thing being this massive log, it's lots of different places. It's not just in that tiny window.
Is it in the other places where it's known? Are they impression fossils? Like what sort of?
Oh no, big chunks.
Big chunks, so they're like kind of like like tree trunks or something. I mean,
we've got massive logs of it,
wow.
But most of the data comes from the internal the internal morphology,
the structure. Yeah, wow, super cool. So also this thing was global in extent. I mean, I don't know, what was the world like in the Devonian? What are we talking about? Tectonic plates? Where was land? Was it all? Were we in a supercontinent?
We got about 3 or 4 big continents.
Okay.
So I don't actually know about the distribution of it. Around the place.
Okay, but so Prototaxites is widely known from Devonian deposits.
Yes.
So it'd be interesting to be able to. Well, I guess we're going back to that thing about preservation and the Rhynie chert having this unique preservation, but going to see whether we have different species, different types of this.
Yes.
Of Prototaxites, but I guess that's very difficult to get out.
Well, it's difficult because you don't have a lot of morphology.
Exactly.
Most of your taxonomy is based on the small series of tubes from nice polished sections of it, which makes life a bit hard.
Yeah.
So let's move on to this final fungal paper. I just dropped it to you in the chat. Have you found this file? So, because this paper came out in January, I thought we needed a little bit of extra something, and also for the people that want to know about the Mesozoic. This paper came out just a matter of weeks ago in PNAS, so Proceedings of the National Academy of Sciences. Fungal proliferation before and after the Cretaceous Paleogene mass extinction event in North America. So I won't spend a long time on this because Prototaxites is what we want to talk about. But these guys looked at some columns in Denver and New Mexico and did some really fine-scale stratigraphy before, after, and during the KPG massive dinosaur-killing extinction event, and looked at the microfossils of fungi in the column, and their hypothesis that the fungi big spikes in fungi might be because of spikes in disease and disaster. So if there's a mass extinction and a lot of things dying, you might expect there to be a lot of fungi because they're eating the dead things or spreading diseases.
Fungi's going to have a field day.
Yeah, I mean, if a planet-killing extinction event comes along, fungi are going to be enjoying themselves to the maximum.
Yeah.
Because they eat dead stuff, right? Just to be clear, that one of the major roles of fungi is that they eat up the dead stuff. So, and what they found was they did find a spike at the KPG in fungi at one of them, but they also found a big spike slightly before it and a slight spike slightly after it. Now, I found this a bit tricky because, of course, if you look in the column, you'll find things everywhere and what you're correlating them with. But they raised this interesting hypothesis that the one slightly before the big dinosaur killing asteroid impact event was potentially related to the Deccan traps, implying that the dinosaurs about 10,000 years before the asteroid hit them were undergoing a massive extinction event before the asteroids left. So there might have been a sort of double tap extinction event going on over there, which I thought was kind of interesting.
I mean, there's been a great deal of analysis on that specific point, hasn't there? And over the years, and I would say that the diversity studies that have been carried out generally fall on the side of there not being an extinction, and that dinosaurs were doing well right up until the extinction interval.
Mm-hmm. As we did when we had our long episode with Steve.
So Steve talked about this a lot, but a large number of other studies have also found that. Now, a minority of studies have suggested that dinosaurs were in decline prior to the KPG, but we have this issue that the record that we have of this time period is almost entirely from North America. And there were major paleogeographic changes going in North America as a result of withdrawal of the Western Interior Seaway, which makes this very complicated. Now, it's interesting because this fungal record could be a totally independent way of looking at it. But on the other hand, it is also from North America. And I don't know, again, I haven't seen this paper. I've just literally opened it. Do they address environmental change and the impacts of environmental change on fungi here? Because I noticed the PG spikes. I'm looking at the figure which shows the fungal spikes, so figure 2, and there's a spike at the boundary, but there's also a spike after the boundary, which is the most profound one. And there's also been some recent studies about how the environment actually changed after the impactor in that part of the world, and suggesting that actually the absence of dinosaurs kind of fundamentally caused like river systems and stuff like that to actually change, the environment changed because these huge ecosystem engineers had gone extinct. And so I think that might be quite difficult to disentangle here as well.
I agree, and that was one of the reasons that we're not doing this for our main episode, I guess, is because I think this is going to quickly boil down to a question about changing patterns of sedimentology or diagenesis, right? So if you're dealing with a huge period of volcanism at the Deccan Traps, and then 10,000 years later you've got a massive asteroid impact, then 3,000 years later you're dealing with a place that doesn't have sauropods anymore, then the pattern of the rivers, the changing of the deposition, that's going to have a big pattern on it. So we're not going to spend ages talking about that, but I think it's quite an interesting idea that and also it tied together with our Prototaxites, as we're thinking about fungi, not fungi, was that they're doing this on a fine scale, and I think that's the way to address this problem. A lot of the diversity studies are like global ones where it's very hard to get a pattern on. This is a really, really detailed sedimentological analysis where you can actually go to the scale of looking not on millions of years, but thousands of years or tens of thousands of years before and after the event, which I think is maybe one of the ways to address this problem. But when you do that, you can't get around the fact that maybe your sedimentology is complicated.
So what you can get around that, actually, what needs to happen now is that we need to do this same study at KPG boundary sections in other parts of the world. So a big problem with that is identifying where the KPG boundary is in some other parts of the world where we have terrestrial deposits. Now, the Gobi Desert is a great place, but we haven't identified the exact KPG horizon in the Gobi Desert. So we know that the deposits are late Cretaceous, but we don't know exactly where the KPG is. But that would be like a kind of independent control, right? If you go somewhere else and then find the same pattern, then those environmental changes aren't going to be the same in somewhere else. I mean, yes, the dinosaur is going to be extinct, but that the impact of things like withdrawal of the Western Interior Seaway isn't going to be affecting the Gobi Desert. So and also it's a bit closer to the Deccan Traps. So maybe it would be, if you see this exactly the same thing, then that's kind of building up a body of evidence to suggest that the pattern is a real pattern.
So in this paper they focus on North Dakota and Colorado, but they also make a big deal of comparing it with a paper from New Zealand.
Right, okay.
And the paper from New Zealand was very focused on the fungi at the actual KPG event. So they are doing that, but I guess now it needs someone to go and find something in the Gobi Desert or, I don't know, in Argentina or whatever, and then go and find the global pattern and see that would help support the picture.
Exactly. But we've got to, little steps are the way forward, right?
Nice. So that was our fungi, not fungi double hitter there. I guess we already covered the take-home messages, which was mind-blowing in the sense that there may be a whole new kingdom of life that we need to figure out. And I don't really know how to do that. My brain is now whirring away thinking, what can you do with that?
Yeah.
How can you test that?
Yeah. Mental.
It's upsetting.
Very good.
I can't even say what you way to go and dig it up. What would you do? No, we don't know. Anyway, Prototaxites. I finally got around to doing it after you asked me to do it for ages.
Thank you.
So
it was good. I thoroughly enjoyed it.
I'm glad you did. Next time, I think we're going to be talking about your Moroccan fieldwork, and then.
Well, that's a subscriber issue, isn't it?
Subscriber. Oh yes.
If you want to hear, I mean, so I did. We did an episode about what I've been doing in Morocco back in probably September 2025 when we announced our spiky dinosaur, Spicomelus, the ankylosaur.
Yeah.
So if you want to find out about what we are doing out there, then tune into that episode if you haven't listened to it before. But we are going to do, I did some live recordings from the field with live digger noises and wind interference on my phone. It's very, very real. It's very real. So if you fancy listening to that, finding out what I did for the last 2 weeks, then like and subscribe, as you say, Rob.
I feel dirty saying that, but yeah. All right, thanks, Susie.
Cheers, Rob.
The Fossil Files was created by Robert Sansom and Susanna Maidman.
Fossil Files is a labour of love created in our spare time with no budget, so we really welcome any support you can provide us via our Patreon.
There you can access bonus content and join in the discussion by posting your own questions or even participate in live episodes.
In either case, we want to hear what you think. You can let us know what you thought about the topics we discussed or make suggestions for future papers in our social media channels.
You can find links to our Blue Sky, Instagram and YouTube channels on our website fossils.libsyn.com.